This is a short presentation on BioSig3D, which is an imaging bioinformatics system for high-content screening of three-dimensional cell culture models. It's a joint effort with Kemal Bilgin, Gerard Fontenay, Hang Chang, Zhou Han, and myself, Bram Parvin. I will start with a few introductory slides and then proceed with a live demonstration. So why three-dimensional cell culture models as opposed to 2D ones for screening therapeutic targets or gaining new insights into biological processes? There are structural and functional differences. In 3D, cells form an ellipsoidal shape with 10 to 30 microns dimension. In 2D, they remain flat. In 3D, cells are exposed to the neighboring cells and the extracellular matrix, ECM, while in 2D, the exposures to the nearby cells are minimum. In 3D colony organization, which is a placement of each cell within a three-dimensional colony, is a clear endpoint for aberrant growth, which may be a prelude for cancer initiation and progression. There are also functional differences. Human mammary epithelial cells cultured in 3D have the capacity to generate milk in culture upon administration of hormones. In 2D, they are not functional. Patterns of gene expression are different, and so are the drug responses. However, in most cases, three-dimensional cell culture models need to be imaged, visualized, and processed in full 3D volume, which impose a number of bioinformatics challenges. What are the questions that we would want to ask from an imaging bioinformatics system? How does the morphometric properties and molecular endpoints of a 3D system are altered as a result of changes in the microenvironment, aberration in the genome, that is mutation or changes in the copy number, or simple developmental processes such as colony morphogenesis? What kind of an imaging bioinformatic platform is needed to support asking these questions? Which brings us to BioSig3D. It consists of several modules that supports a laboratory information management system, a LIM, 3D visualization of raw and processed data, analytical engines for image analysis, and bioinformatic tools. These components are brought together through the strong use of controlled vocabulary. The system has been validated with 4 cell lines with diverse genetic background that were cultured in 3D and then harvested over time. Here is the website for BioSig3D, which includes documentation and installation manual. With that, let me give you several views of the BioSig3D. When the user logs in, he observes a number of studies. For each study, there could be a number of experiments. One such experiment is shown over here that consists of 2 experimental variables, cell lines and harvest time. So collectively, there are 16 distinct experimental variables. Samples are then imaged and collected and linked to these distinct experimental variables. Here we show, for example, this row demonstrates the samples that have been imaged corresponding to MCF-DNA harvested on day 12. Only the middle slice of the colony, a three-dimensional colony, is shown in this picture. Each sample is then processed, that is segmented in terms of its nuclear structure or cellular structure, and then molecular measurements are made. More importantly, we also measure colony organizational features, that is the placement of these cells with respect to each other in a three-dimensional colony. If enough samples are collected, then we can proceed with a bioinformatic analysis in terms of the heat map. So every row corresponds to a distinct set of experimental variables, and every column corresponds to an index that is computed as a result of morphometric property, molecular endpoint, or colony organization indices. With that, let me proceed with the live demonstration. Here's a website for BioSig3D. When the user logs in, he observes a number of experiments. He has full access control in terms of sharing these experiments or keeping them private. So I will open up this particular experiment, which involves colony morphogenesis. It has 2 experimental variables of cell line and harvest time. And here's the combination. And the user can annotate which set of experimental variables are controlled, which one are treated. In this case, the sample have been stained with DAPI and actin fluoride, And the user can then proceed with the plate design in terms of which experimental variables or distinct set of experimental variables are placed in a particular well in a plate. The data are then collected and linked to these experimental variables to these via this tab, the image transfer tab. We can then proceed with visualization. So here are all the data that have been collected as a result of this particular experiment. And if I click on any of these thumbnails, I can see the full 3D volume from the top to the middle to the bottom. We could also look at segmentation result. The system supports multiple policies for visualizing segmentation result, which is essentially for quality control. The default is JavaScript, and if the user has a graphic card, he could also use WebGL. So this process moves a little bit faster with the WebGL. So as a result of this operation, nuclear morphometry is computed. Molecular endpoint for each nucleus is also computed if the samples are stained, and colony organizational features are also computed. We can then proceed with bioinformatics analysis. We support two different policies in terms of mean analysis or distribution analysis. And I will randomly select a few indices over here, which corresponds to colony organizational indices and subcellular morphometric properties. And the system dynamically computes the a heat map based on the experimental variable, which is every row, and compute selected indices that were computed as a result of the processing. We also support a number of other bioinformatic analysis in terms of correlation of the colony features to cellular features, and also enable export of computed features to a local desktop for further bioinformatic analysis. This concludes our presentation.